So in this project, we worked with soybean. We worked with 4 varieties of soybean, 2 conventional and 2 transgenic varieties. One variety that just have one type of insertion, like cry 1B. It is a protein that the insect eats the leaves and die. And another soybean have 2 different insertions to resist, like Professor mentioned before, to resist the herbicide application, and the cry protein insert also. So in this project, we worked with dot, seca, dry, draw, and we verified that when the plants have many stressful situations, these plants expend many energy, like carbohydrates. The pathways that we find like disturbed was carbohydrates, the relationship with microorganisms, and susceptibility to have disease. And we also check that the plants that have 2 insertions are more susceptible than the plants that just have 1 insertion. So these plants have more stress when subject as stressful situations. Was basic this defines, and we check also the grains after the complete season, and we verify that the grains have residues of glyphosate pesticide. So we just sprayed 1 time, and we verify in the final of the cycle of the soybean a huge percentage of pesticide doses. So and we also check the composition analysis, but we don't find not substantial difference. We just check the pathways and the pathways, and we check these many pathways disturbed around the transgene insert. Yeah, I think that the science advance in this way. We have many techniques that we can check the effects provided by 1 insertion in the in the total metabolism on the pathways that maybe can have a change. So I think that the companies, or maybe the laws and the governments must be run in this side, evolute how the science evoluted, check with the omics, like the omics, we can check how is the effect provided by 1 insertion. We can check this. We can check this because we have sequence, we have proteomics, we have metabolomics, we have many compounds that can be changed by just 1 mutation. we have many allergenic compounds, and we don't know where these compounds come. So maybe if we have the holistic vision and check the totality of the biological system, we can move forward of this issue. Because many researchers are financed by these companies, and so they can find something for these companies, but our search is in another way. We are like unbiased science, and we check because our search is in the side of biosafety. So our concern is check what is the effect caused by these techniques, because it's our food. So we are concerned by this, and this food feed many organisms in environmental, so, and interact with the soil and water and air and insects and another lives. So I think that it is important to have this overview, this broad overview of the science, and we don't work with one product. We work with a life, a cell, and this cell can transform itself to survive in a diverse condition. We see this with soybean. When we search soybean in stressful situations, we see that soybean move your body, yourself to have the fitness to survive and provide seeds to live in the stressful situation. So I think that biology is one ecosystem that was influenced by the environmental and interact with humans. And in the side of CRISPR, we have this problem, because DNA is just DNA, but enzymes that we put in the system can interact with humans and animals that we eat, and another food that is secondary for their life, like trees or birds. Yeah. So the footprint in this context is like the same view. We have the overview of the science, the introducing a mutation, but when we work with CRISPR, we are more specific in, I can say specific, in genome. But in the same way, the scientists can work with the old techniques like vectors, bioballistic, and these effects can be very random in the genome. So we have a new technology, but the old problems, the random effects in genome. So the proteins and the products produced by genome maybe are changed, and in Foodprint we just have now we work it with the detection. So we try to to detect mutations, single mutations, because all GMOs have many structures of genomic, like a promoter, stop codon, and other things. But we can provide one genetic food just with a single mutation. And how we can detect this single mutation if you don't have a promoter? Because in the older GMOs, we can detecting just by a sequence of promoter. We make a PCR and we see the bands in gel or in the qPCR. We can attest that this product is positive, have GMOs. But in this context, we are in num terreno lodoso, como é que eu posso dizer? Num terreno difícil, in one difficult land, because we don't know how we can detect one single mutation. So we can try figure out this, and we will try many techniques in qPCR to try specifically detect one single mutation and prove that it's possible or not identify this product. I think that Brazilian people just don't know that these mosquitoes are in the environmental. Must be that the São Paulo people don't know that the mosquito living with there. So the information about this is the first thing that the researchers or the government must be informed, the people that live there. And another thing is that the CTN-Bio, the Brazilian Commission, have a huge role in this situation, because they are able to read and have the risk assessment about this mosquito. So we don't know, we don't listen about this. So how we can move forward to search about the risk of these mosquitoes? We don't know. We don't know the egg. How we can discriminate the egg from the transgenic mosquito to the conventional mosquito? We don't know. And the mosquito interact with the flora, like bromelia and another native species. So we don't know how this can interact with another species, or the birds that feed this mosquito, or the frog that feed these mosquitoes. We don't know. We just don't have knowledge about this. Just, for me, sounds like a testing, population testing, like a tentative to eliminate mosquitoes, but we don't know. We know that this is like a fail, because we have many historical situations in agriculture that we can prove that the evolutionary move forward, and the evolutionary pressure make the organism more capable to survive of this situation. So probably in the future we have like a resistant in the two like a huge mosquito that we don't know how this mosquito can circulate and interact with the environmental. It's for me it's like it's a big concern, because if we did this with mosquito, we can did with another insect. And in agriculture we have many pests and we have many fungus that are a problem. So maybe this technique can be replicated in agriculture, and maybe we can feed with food that have this technique and we don't know. Ah, it's a difficult question. I'm arising in this land maybe 8 years, but now I am I have more knowledge about these legal questions and how the society can provide their own solutions for their own problems, like Professor mentioned before. But in the biosafety view, I believe that when we have a company that controls the technique, it's difficult that we, like good science, move forward in the same velocity. Like we just walk enquanto, I forget the word, while the industry running. So it's difficult in the biosafety view, because sometimes we don't have the background to comparate like the conventional varieties to the transgenic varieties. So how we can see the changes? We don't have the background, so this is very difficult sometimes. And in the mosquito, I cannot imagine how the biosafety can provide the changes, because it's difficult to discriminate the mosquitoes transgenic that don't transgenic. So I think that when we work with plants, it's more easy, because we can collect the seeds when we have. But when we work it with insects, it's very difficult, or maize, like you mentioned before, because we need to consider the reproductive system that the plants have. Maize is very promiscuous. We have pollen, the flow of pollen, it's very huge. So we don't can dimension it, I think. We cannot make a like a box and infer the things in this box, because when we think in science, the system is more big than the box, and when we infer just in this box, we forget the other things that interact with this box. So I don't have the answer for your question this last, but I think that the precautionary principle is in the table, just because this por causa desses casos, né? Because of this case, we have regarding the CBD and regarding this principle. When we don't know the risk, maybe we need stop and search more about the risk. It is international project, yeah, and our part is work with detection and based on PCR and in off-target effects, and also in target site. So we have partners in Lebanon and Austria that move forward with us in this approach to detecting, and they will validate our methodology in qPCR. And we work now with Arabidopsis, a model plant, to try detect the mutations provided by CRISPR-Cas9. And also we developed one platform to make edition in protoplasts in soybean, and we have a model for do the edition in protoplasts. So this protoplast in next year we will evaluate the off targets and maybe propose one detection overview based on multi-target approach. I believe and I see that is very weak in this moment, in this historic moment, because our commission is very biotech oriented. So we don't have more discussions, more society discussions, and the ONGs that bring these discussions for society and the commission are like, so this discussion is very weak, and discussion for gene editing less. So I believe that this moment is very important, mainly for Europe, because Europe export like many manufacturers products that Brazilian export. So maybe in Brazil the situation and the advance in the forest for planting this soybean transgenic or gene editing can stop by the international pressure. But here I don't believe that the situation can change, because we are an emergent country, and this experience arrive like in the countries like Brazil, Argentina, inputted by Estados Unidos. So I believe that here we have a weak discussion, because we have this view that biotech is very important for feed the world. But biotech is cotton, soybean, and maize. We have a huge forest that can provide many foods for the world, and another components that can be explored. with the society, with the indigenous people. So I believe that here in Brazil, the situations cannot stop by own, just because the international pressure. Maybe we can bring this discussion, this CBD discussion again, is weak. we can bring this discussion more strong, because the climate change, the food change, the perspective of future change, so we can bring this conservative vision again, and one overview based on this synthetic biology is very important.